To know our world and ourselves, science is the means. To discern how science progresses, breakthroughs are the ways. There are tectonic breakthroughs shifting whole fields of science, like quantum mechanics and relativity in physics, evolution and the genetic code in biology. There are incremental breakthroughs, the quiet, small steps science makes. But the substance of breakthroughs, the content is one thing. How breakthroughs happen, the process, is quite another. How to explore the process of breakthroughs in science. Are there principles or commonalities that underlie all breakthroughs? Physics drove breakthroughs in the 20th century. Biology drives breakthroughs in the 21st century. Biologists work in different worlds than physicists, separated by vast orders of magnitude, numbers of variables, and levels of complexity. Does the process of breakthroughs by biologists differ from the process of breakthroughs by physicists? How do breakthroughs happen in biology? I'm Robert Lawrence Kuhn, and Closer to Truth is my journey to find out. When biologists reflect on how they do science, when they consider all they do, all they endure in making their breakthroughs, small or large, all the false starts, hesitant steps, blind alleys, what do they think? How do they feel? To explore the process by which scientific breakthroughs happen, I speak with biologists who have made breakthroughs from plants to neuroscience. What are the building blocks of biological breakthroughs? I begin with a visionary physicist who pushes boundaries in biology, Paul Davies. Paul, I'm fascinated by the breakthrough process, and you've been involved in many, in physics and black holes, astrobiology, cancer research, and origin of life. What I'd like to understand is the process by which you've gone through.
When you look back through the examples in history of major breakthroughs, they really come about for two reasons. One is the discovery of something new, experimental or observational discovery, that this upends existing thinking, like the expansion of the universe, for example. The other type of breakthroughs occur because of a conceptual mishmash. A mentor of mine, John Archibald Wheeler, the great gravitational physicist, liked to say that progress in science comes more from the clash of ideas than the steady accumulation of facts. And so if you have one way of thinking about a particular phenomenon, and then a second way of thinking about it, and there's some sort of disconnect, then that usually means that we're either asking the wrong question or we need a new conceptual scheme to come up and rethink the whole field. And it's the latter sort that interests me, because I suppose I'm a concepts person, and I always think that if I can somehow reconfigure a subject or reconceptualize it and come at it from a different direction, that is likely to result in a really major new way of thinking. When Stephen Hawking produced his famous result about black holes not being black but emitting heat radiation, there was the mystery about how does the energy get out of the black hole. It was supposed to swallow everything. And that required thinking about the nature of energy in a completely new way. It was a way informed by the theory of quantum fields, as opposed to our naive view that a little blob or region of space would have some energy in it. And so this was, for me, an important way of just thinking the problem backwards. And thinking backwards is actually often a good way to come at something that you tend to have a sort of linear chain of reasoning. In recent years, I've become interested in the crossover between physics and biology. If you ask a physicist, what is life? You'll be given an answer in terms of forces and molecules and interaction rates and entropy and those sorts of physical type things. Ask a biologist, what is life? You'll be given an answer in terms of information, of signals and coded information in DNA and patterns of information flow in gene networks and signaling between organisms and so on. And completely different way of thinking about the same subject matter. And so that to me, suggests that we need to redefine, reconceptualize life. The question, what is life, which was asked by Erwin Schrödinger 75 years ago, remains a fundamental problem in science. And I think the answer to that will only come from coming up with some new type of conceptual scheme.
Not everybody agrees that that is a potential breakthrough, because they think that physics in a reductionist manner just obviously would explain biology.
Right, and so among biologists, there is a tendency to think, well, it's all incredibly complex, but sort of deep down, it's all just physics operating at the level of atoms and molecules.
Right.
Now, I'm not saying that life is anything beyond physics. I think life can be explained by physics, but it can't be explained by known physics. I think we need new physics. And in order to come up with new physics, we need a new way of thinking about the nature of life and a new way of thinking about physical law. So I'm suggesting something very radical.
Paul highlights two drivers of breakthroughs: a discovery, a new experiment or observation, and a conceptual discrepancy or contradiction. But breakthroughs in biology must recognize that biology is not physics, and that reductionism to current physics won't work. We need a new way of thinking about the nature of life. What could constitute a new way of thinking? Could one way be recognizing hierarchy levels in biological explanation?
I mean, not just
I explore with a neuroscientist who has made affect, emotion, how we feel, central for understanding cognition, Antonio Damasio.
I remember as a young man thinking a lot about affect, but always in relation to the arts. See, I was always very interested in movies. I was always very interested in theater. And that world was sort of a compartment where I thought affect was fantastic. Affect was telling us something very important. But when I started doing scientific work, what I thought about was, in fact, cognition. And almost everything that I did early on had to do with language, which was a main interest of mine. And it was only much later, largely forced by observing certain neurological patients with certain conditions, that I was sort of dragged, kicking, into the world of affect. So actually, on one given day, I made that change, and we had an agreement, everyone in the lab. There were several people participating in this, and said, we're going to make a change here. We're going to give this topic the priority.
Wow. All right, then I see these other critical steps that you've made, where affect then creates mind, and then mind to consciousness, and then consciousness to larger implications, both evolutionary and to culture. Were those equally as discrete, or is it just sort of a natural process?
I think I have a little bit of clarity as to how I see the beginning of minds in evolution, and how I see the connection between affect and consciousness, and particularly feeling and consciousness, because the two are absolutely joined at the hip. If you ask me what came first, feeling or consciousness, the two came together. You cannot have a feeling if you are not already conscious, capable of being conscious of that feeling. It doesn't make any sense. To feel is to be able to connect a particular state with yourself, with the me that is behind that process.
And then you go from that to a broader understanding of human culture and human civilization.
Correct. It's a sort of rite of passage. You have to have that joint between consciousness and feeling, and then the world can open up. Is this distinction between the feelings that have to do with our body, with ourselves, which we can describe technically as homeostatic feelings. For example, well-being, pain, suffering. Those are feelings that have to do, for example, being thirsty or hungry. All of these are feelings that are expressing states of the organism and giving a mind a very clear indication as to whether you are in the correct range or not, and pushing you in a certain direction. And then there's something else that develops, I presume, much later, which is the kind of feelings that we relate to emotions, the feelings that express pleasure or anger or fear. Those are of a different nature. They don't have to do with the regulation of an organism primarily. They have to do with the regulation of an organism in a social setting where others are present as well. And they relate to the connections we make or do not make or badly make with others.
For Antonio, his journey began by recognizing that the split or distinction between intellect and affect, emotion, while natural, was artificial, a barrier to deep understanding. Antonio made innumerable small steps in surmounting the barrier, culminating in his career-defining big step, a redirection to focus on affect, thereafter studying affect's grand impact on consciousness and culture. But must impact be grand to catalyze breakthroughs in biology? What about anomalies, small variances, or abnormalities? For example, clinical cases of brain damage from trauma or disease. Neurological problems can be discrete, even odd. I explore the process of discovery while studying patients who have strange conditions such as phantom limbs with a pioneer in the field, V.S. Ramachandran.
Well, you have to introspect on what goes on in my mind, and that's not easy. But usually one has to be wary. I mean, all these people select me, and they sort of phone me and say, you're interested in strange syndromes, and we have a patient with such and such, please. But in the beginning, how did I do it? I'm not quite sure, but I can tell you this, that a lot of them turn out to be wild goose chases. You don't hear about the failure.
Right, that's a good,
And I
very
can
good point.
mention a couple of those, just for fun. One example is Duclarenbold syndrome. The diagnosis of the disease is based on a young woman developing a delusion that an old, famous, rich guy is madly in love with her, but is in denial about it.
Right.
Okay, it's crazy. Now, the converse of this would be where an older guy develops a delusion that a young hottie is in love with him, but she's not. And that's much more common.
Yeah, that's right.
Because it doesn't have a name because diseases are named by male psychiatrists.
So what's the implication of that?
That you have to be very careful in pursuing odd syndromes. A good way to make new discoveries, but it's also a good way to get misled. So the reason I mention this is there are countless examples of this in neurology and psychiatry, and you want to make sure you're latched onto the right Kuhnian anomaly. The anomaly, I mean, everything is the edifice of science, but something that doesn't fit, which you think shouldn't be allowed, and you sort of brush it under the carpet. But every now and then an anomaly reaches a threshold, and you can't ignore it, and it says, I'm part of the story, you know. So you have to tear down the whole edifice, start from scratch.
But you have to be careful.
After some years, you get a nose for anomalies.
Yeah.
Which ones are valid, which ones are not. Usually, if a phenomenon has been repeated in a laboratory, one example is what's called the calendar line. Right? And where people are asked to imagine the annual calendar, they'll say January, February, March, April, and then down to December in the lower right. And it's true of most people. But 1% of the population see the calendar literally. Well, I actually see the font and the black outline around each month, floating in front of me in space, idiosyncratic shape. Each person is different, or it's an L shape or a V shape, with very specific letters and defining the calendar. We were able to show, first of all, it's a real phenomenon, but doing simple psychological tests. A normal person finds it very difficult to name backwards or name alternate months backwards, but a person with calendar synesthesia does it effortlessly. But he just reads it off. You and I can't do that. Now, the question is, where does it take place in the brain? And the answer, I think, is there's a structure called the angular gyrus in the left hemisphere, which is involved in, we think is involved in sequence. The calendar, after all, is sequence, January, February, March. And there's a fibers going down from the angular gyrus down to the hippocampus, which is the memory center in your brain. It plays cells and groups of cells in the hippocampus which are involved in orienting in space and time. I am here now, I get up in the morning, I'm here now in this hotel, I'm looking through the window, I'm in Los Angeles, that sense of perspective. That you link that to the calendar, and I'm supposed to get up early, I'm supposed to go and go and talk to these people at this meeting. So this whole process is occurring in this band of fibers and its connections. Getting to that level of precision, studying a patient, is very satisfying. What we do for a living is look at anomalies, look at patients with sustained injury or change in the brain, manifesting strange, odd behaviors which are inexplicable, then play Sherlock Holmes on these patients, and then find out what's gone wrong in these patients. But the really hidden agenda is find out how the normal brain works, not merely explaining to the patient what's gone wrong with him, plus find out in terms of neural nitty-gritty brain tissue, what's going on in the brain tissue that's causing this behavioral change. And finally, can you do something to help him? In the case of synesthesia, you don't need to. I mean, he enjoys his calendars. But in the case of phantom pain, for example, you need to fix it.
Right.
You don't hear about the failures, Rama says. You get a nose for anomalies. And when an anomaly reaches a threshold, you can't ignore it. You must tear down the whole edifice and start from scratch. Failures as guardrails, anomalies as signals. I like that. Most of biology, of course, works quite apart from human beings. How do breakthroughs happen in basic biology? I meet a leading plant geneticist, credited with critical breakthroughs, Daniel Chamovitz.
I think the scientific process in general is one, first, of the scientist himself being aware and making connections that he hadn't thought or she hadn't thought of before. I started asking the question quite randomly, actually. Why do plants make chemicals that affect human biology? From an evolutionary point of view, it doesn't quite make sense that you make something in case someone eats you. There has to be some type of reasoning for that. And so in my laboratory, what we did, we just bought one of these chemicals. It's a chemical called indole-3 carbinol. It's part of the taste of mustard, and threw it on the plants. And what we found out is that these plants stopped growing. Now, if you think about it, when people talk about mustard and broccoli, that's supposed to be protective against cancer. And when we added it to the plants, their cells stopped dividing, which was what you would want for an anti-cancer drug. But now comes the scientific process. We started asking, how is this chemical working in the plant? It's not doing it to stop cancer in humans. And through a series of experiments, what we discovered is that the plants use this chemical as a signaling molecule to let the plant know that it's been under attack by an insect. In other words, when the plant was attacked by an insect, it makes this chemical. The plant then stops growing, the cells stop dividing. The plant then uses its resources to fight off the insect. When the insect has gone away or when the damage is gone, then the chemical goes away, and then the plant starts growing again. So the same chemical which we could use as a drug, the plant is using for an internal signal.
And what are some other examples?
The way I got into studying plant senses came from my postdoctoral research at Yale University when I was interested in how plants differentiate between light and dark and use this as a signal for development. I was actually looking for a question that was plant-specific. But through my research, I discovered that the genes that I had found that help a plant know if it's in the light and dark are also found in the genome of every animal and every human, which then led to the question, why would humans have these plant-specific genes? And then led me to the realization that the distance that I'd mistakenly thought between plant biology and animal biology doesn't exist, that there's an incredible unity in the way that all organisms respond to their environment.
What's the percentage relationship between the genome of plants and animals? Order of magnitude?
40, 50%. Maybe not surprising because we all evolved from that same single cell, which was in the oceans several billion years ago, which evolved to yield both animals and plants. And so any gene that's necessary for cellular life, of course, is going to be found in all organisms.
And the survival mechanisms?
These same genes that I found in Yale when I was a postdoc help a plant know whether it's in the light and dark and modulate its development. In humans, these same genes are involved in developmental processes which, when go awry, lead to cancer. So you're going to get tissue-specific responses, which are different in plants and in animals, but the basic cellular biochemical mechanism will be the same. The implication is that by studying plants, we can learn a lot about human biology at the basic cellular mechanism. And this comes back around to why plants make this variety of chemicals, which we then use in medicine or as drugs. Because these same chemicals in a plant affect plant biology, so of course they would also affect the shared human biology.
Though plant biology may seem a world apart from human biology, realizing their common ancestry enables new insights. That's the key connection that drove Danny's breakthroughs. Sometimes strange connections yield breakthroughs. I meet mathematical biologist Leon Glass.
Leon, I love trying to understand how scientific breakthroughs happen. So I'd like to ask you about glass patterns, which, not coincidentally, is your name.
I had done a PhD in chemistry in Chicago. I studied motions of molecules in simple liquids. But I was interested in the brain and wanted to go study someplace where I could study the brain. I went to the Department of Machine Intelligence in Edinburgh. And my mentor there, Christopher Longa-Higgins, suggested that I start studying visual illusions. And he said, here's a fantastic effect. We can take a blank piece of paper, we can take a Xerox copy of that blank piece of paper, and then we can take a Xerox copy of the Xerox copy. And then we can take a Xerox copy of the Xerox copy and the Xerox copy of, et cetera. So you can keep on iterating this process just with starting with the blank piece of paper. Now in Edinburgh, the Xerox machines had a lot of dust in them and the paper had a lot of little pieces of brown stuff in them. And after you did this about 10 or 15 times, you got a pattern of dots on a piece of paper. Well, I was interested to figure out if these were random or if these were regularly distributed. So in order to study the pattern of those dots, I made a transparency of the dots to project up on a wall, and I made a bullseye pattern. I was going to put one dot in the center and count the number of dots in each ring. And in the process of doing that, I noticed that when you superimposed one set of these random dots on the original, in a little translated position, you saw circles. If you rotated the transparency too much, then the circles disappeared. So it had nothing to do with what I was investigating, which was the spatial patterns. But Christopher Longa-Higgins looked at that and said, what's happening there? Why do we see circles? So that was the discovery of these moire patterns from random dots, which people have started calling glass patterns.
Is a glass pattern something that we're projecting onto it, or is there something there really that's...
It's both. It's both. With everything, there's something there.
Right.
And there's the way the brain figures it out. In some cases, if you rotate one of the sets of dots too much with respect to the first one, you can no longer see it. There's still correlations there, but our brain can't perceive them. It's not immediate, it's not striking.
Right, so it has to be within the range that the brain can expect to see.
Exactly what the brain is doing is something that I think people still don't know.
But it is doing something.
For sure.
Everybody used to just think it was just sensory going up and then processing the brain. But the brain has a lot of efferent fibers going down to the sensory organs, so it puts a predictive pattern that it expects and therefore amplifies things.
So exactly, the initial paper that we wrote was based on work by Hubel and Wiesel, who describe how simple cells in the visual system responded to straight lines. So the notion was that these dots would form lines in different directions, but there were more in the correlated directions in any area. So the initial paper suggested ways in which the brain could do the computations, which would be underlying that process. The extent to which things are coming from top down, where the brain is saying, well, I expect to see, this is one of the things I can see.
Would have been some applications of glass patterns. But in terms of understanding how the brain functions.
My feeling is that even in understanding still very simple processes, we are still very far away from understanding what's happening.
What is the process of breakthroughs in biology? There are no chemistry set paint-by-numbers directions to follow. What works is to experience breakthroughs vicariously, live through diverse thoughts and emotions of those who have made breakthroughs. Breakthroughs are driven by new discovery or conceptual discrepancy. Try thinking backwards. Many small steps can transform into a giant step. One breakthrough can lead to others. Failures abound. We don't hear about them, but appreciate them. Get a nose for anomalies, tear down the whole conceptual edifice and start from scratch. Though scientific fields may seem worlds apart, find deep connections or common ancestry. Follow odd observations, even if outside of your field. Breakthroughs in biology are not formulaic and not mechanical, nor can they be legislated by fiat. Breakthroughs can be stimulated and encouraged, but not ordered or coddled. Breakthroughs are milestones and touchstones. Without them, forget ever getting closer to truth.
For complete interviews and for further information, please visit closertotruth.com.